That would be good. And so just a couple things to get us started, and then I'm going to pass it over to my colleague, Brianna, who's going to introduce our keynote speaker. So just a couple Zoom etiquette reminders for you all. This will be the link for the entire meeting today and tomorrow, so you can click on that to rejoin the meeting at any time. If you can, go ahead and put your full name in it, first name, last name, or anyone you're letting know coming onto the meeting. That helps us know who's here. And if you have trouble getting on the meeting, you can email Steve Elmer with your phone number and he can call you in. So he'll put his email in the chat for you shortly, and you can do that. When we're doing the main session talk, so this keynote talk and some of the trainee ones later, if you could have your camera off and mic muted, that would be great. But we would love it if you would turn it back on during questions, during all of the breakout sessions. We want to see everyone's face and make this as interactive as possible. And so I just want to give you a quick overview of the schedule for today, and then I'm going to pass it over to Brianna. So as we see here, we're going to start off with this really exciting keynote lecture by Dr. Esser. At 12:30, we've got our undergraduate trivia competition with Kahoot. That was very fun last year, so looking forward to that again. We'll have a little bit of time in between there just for a social half hour, and we hope that you all can stick around and chat just as we were before the meeting. It would be great to catch up with everyone. From 2 to 3, we'll have 4 of our amazing trainee presentations. That'll be a main session talk, so everyone all in the same room. And then at 3:15, we've got some really great breakout sessions scheduled. And you'll see the titles there. So hopefully something that can interest everyone, and you'll get to go to any 2 of those rooms during that breakout session. So as I said, lots of exciting stuff coming up. And we are very excited to kick it off with our keynote lecture. So I'm going to give the floor to my colleague and the treasurer of MPS, Brianna Harfman.
Thank you, Jenny. I am very excited to do this introduction. I actually asked this introduction because Dr. Karyn Esser was my PhD mentor. So it really is a true pleasure for me to introduce our keynote speaker today. Just to give you a little background, Dr. Karyn Esser did her training, her education at Wake Forest and UNLV, culminating in a PhD at University of Michigan, for those of you who are from U of M in the audience, and a postdoc in Sydney, Australia. She began her career at University of Illinois, Chicago, and moved from there to University of Kentucky, where she also served as director of the Center for Muscle Biology for a time. She's currently at University of Florida, where she is a professor of physiology and functional genomics and associate director of the Myology Institute at the University of Florida. Her lab has been working in the area of skeletal muscle adaptation for over 20 years. And initially her research was focused on understanding the molecular mechanisms that underlie adult skeletal muscle adaptation to exercise. However, in 2002, she accidentally discovered that genes important for circadian rhythms were also at work in skeletal muscle. Since then, Dr. Esser's lab has pioneered research on the role of circadian rhythms and the molecular clock mechanism in skeletal muscle. And we're going to hear a little about that today. So I'm going to turn it over to Karyn.
All right. Thanks, Brianna. And I want to thank everybody. Let me make sure I get this set up correctly. Are you seeing it? Does this look appropriate now?
Yes.
Okay. All right. All right, great. Well, again, thanks everybody for, and thanks to those that invited me. This is a great opportunity to share some of our work. And yes, the state of Michigan and the University of Michigan are very fond memories for me. Anyways, what I want to talk about today are really primarily muscle clocks, but then some of the work that we're doing that is starting to unfold some interactions with exercise, with some ideas about how this might be useful therapeutically or strategically. So now that we're in the era of Zoom, that I think it's pretty well understood that if I sat here and talked to you for the next, 45 minutes on one thing, it would probably get a little old. And so what I've tried to do is break up this talk into 3 short stories. The first one is going to be talking about sort of getting everybody up to date, up to speed with the concept of what are circadian rhythms and what really are circadian clocks. So this is a field that has been rapidly emerging. So rapidly in my time frame, but in terms of biomedical science, and it has applications for lots of things related to physiology, but also in terms of therapeutics and disease and aging and all things, basically, as far as I'm concerned. Then the second short story will be really more specific to the tissue that I've been most fond of over my career, and that's skeletal muscle. So what are the circadian clocks doing in skeletal muscle? What did this accident lead me into? And then the last topic is going to be some work that we've been doing in the lab, looking at the interaction between muscle clocks and exercise, in particular time of exercise, and how they might be talking to each other and what this might mean either for therapeutics or athletic performance. Okay, so to get started on this, so what are circadian rhythms? I'm going to assume that everybody in the audience actually is familiar with the term. So circadian rhythms are well known. They're basically this concept that there are repeating approximate 24-hour biological events. So things like sleep-wake cycle are what we are most familiar with. But I think it's really important to point out that circadian rhythms are part of biology on planet Earth. So this is not just a human or a mammal thing. This is across animals, it's across plants, and it's across, it's seen in bacteria and other organisms. Now, one of the key features of being officially called a circadian rhythm is that these rhythms that are occurring have to happen in the absence of any environmental cues such as light. So again, if we stick with the mammalian system, we think about light, dark, we think about, it gets light, we wake up, it gets dark, we go to bed, that kind of thing. The reality is we don't need light, dark to have these rhythms. These rhythms occur in the absence of these environmental cues. This is a critical feature of circadian rhythms, in part because it means that these rhythms are actually coming from within the organism or within you. So again, and then the other part where this field has really exploded is that classically circadian rhythms were defined by behavioral patterns, as I said, sleep, wake, or rest activity cycles. But now circadian rhythms have been demonstrated at a lot of different levels, and these levels, physiology, so heart rate changes, blood pressure changes. There are transcriptional changes that are driven in a circadian manner, epigenetic changes, proteostasis and metabolism for sure has very circadian aspects to it. So if anything, what I'd hope you'd walk away from this talk understanding or appreciating that circadian rhythms are actually just a fundamental part of all biology. So this is going to be something that's going to be there. Now, what I want to do with this slide is I want to talk about what is a circadian pattern, or what does circadian behavior look like. So what I'm showing you is some data from a mouse. This would be these are called actograms, and hopefully you can see my pointer. So at the top there is the dark light bar that goes 0, 12, 0, 12. So this just highlights, so this is data from a single mouse that's in a cage, in this case that has a running wheel. And we have the mouse that's living under 12-hour light and 12-hour dark conditions, which is what a lot of animal facilities have their mice on. Mice are nocturnal. So what that means is they're active during the dark phase. And then they get most of their sleep during the light phase. So what you're seeing in this top plot is, and then along the sort of the y-axis here are individual days. So day 1, day 2, day 3, day 4, day 5, day 6. So what you're looking at for any one row is the behavior of this single mouse over the course of 2 days. All right. And you can see that when the lights go off, the mouse gets active, gets on the wheel, starts running. That's what all the black lines are. In the afternoon, or what it would be, it's afternoon, it takes a break. Maybe it's a nap, maybe it's eating, we don't know, but it's clearly off the wheel, gets back on for a little late afternoon running, and then just gets ready and sleeps during the light phase. So again, this is a normal pattern for a mouse and under light dark conditions. However, they are under light dark conditions, so we can't refer to this as being circadian. This is just a diurnal pattern that is exhibited under light dark conditions. So what happens to test something in terms of its circadian periodicity? We have to put the mouse in what we call DD, or basically constant darkness. So, and hopefully that's obvious here with the gray across all times of day. So what I'm hoping you'll notice is that even though there's no light dark for this mouse maintains a rhythmic behavior. So it gets up, it gets on the wheel, it runs, it finishes its running, it eats, and then it'll rest, and then it starts over again. And so what you're seeing right now is a mouse that is exhibiting circadian activity rest behavior. So there's no external environmental light cue. The food is ad libitum, the lights are off. This animal's just rhythmic because it's intrinsically driven. Now, one of the things you'll notice is that, and it's sort of depicted with this red line, is the mouse actually wakes up a little bit earlier each day. And so, and then it'll start running. So it gets up about a half an hour earlier each day. And that's maintained throughout the period of time in constant darkness. So we can use the slope that gets generated by this earlier awaking, and that's what the red line is. And we estimate that the intrinsic circadian period length of this mouse is a little under 24 hours at 23.7 hours. All right. So this is, and if we did this with a human, so let's take, we take one of you, and let's take Brianna, and we're going to take her down to Mammoth Cave in Kentucky, because once you get in Mammoth Cave, there's no light, the temperature is constant, and actually people have already done this, so Brianna doesn't need to do this. But what happens when they live in that cave is they have greater than a 24-hour rhythm. So people are rhythmic, their heart rate is rhythmic, their blood pressure is rhythmic, their wake activity cycles are rhythmic. But instead of, like the mouse is a little less than 24 hours, humans generally are a bit more than 24 hours. And so because of those behaviors in constant conditions, we can conclude that these animals are circadian in nature. So this is another, I talk about heart rate and blood pressure, but I, one of the questions is, because I'm a muscle person, I really like muscle. Did you know that human maximum isometric strength exhibits a time of day difference? And actually this has been studied for many years. So, I mean, really going back, trying to remember when some of the earliest papers were in the early 2000s. So this has been studied for almost 20 years now. And in the figure that I'm showing here, these are a cohort of males, college age males, in which and these are the same subjects, and they had them do 1 repetition maximum isometric contractions at either 8 in the morning or, 8 in the morning or 6 in the evening or afternoon. And hopefully what you can see is that the strength is up to 12% different in the 6 p.m. measurement versus the 8 a.m. measurement. And this has been, so in this case, this was quads, but this has been seen in elbow flexors. It's been seen in grip strength. It's been seen in males and females. And so that there is a very strong indication that there is a time of day difference in strength. And whether now and getting back to the circadian definition, I mean, this has not been measured under constant conditions, so we can't officially say that this is a circadian change, but one would hypothesize that this is probably related to some kind of circadian changes in the organism. All right, so we know this is happening. So then the question is, how? How does this happen? What's the mechanism that underlies these circadian changes in rest activity cycle, or circadian changes in strength, or, like I said, heart rate, blood pressure? Well, the 3 men in the middle, the 3 photos of these men in the middle, so Michael Rosbash, Hall, and Michael Young, these 3 guys were awarded the Nobel Prize in Physiology or Medicine in 2017. Because they had done work in the 1980s where they had used flies that exhibit circadian periodicity to discover the genetic mechanism that underlies this circadian rhythms. And so not just the genes that were involved, but how the mechanism worked. So this, the Nobel Prize was awarded in 2017. And so the work in flies was ongoing in the 1980s, but for perspective, the research that actually reported the first mammalian clock gene, or the first clock gene in a mouse, was published in 1997. And then the first discovery of the human version of a clock gene was published again in 1999. So while this may seem like a long time ago, for some of us, this is not as long. So this is, one might view this as somewhat recent discoveries. So what's the circadian clock? Well, it's a transcription-based timekeeper. And so there's a couple of points I want to make with this slide. So what the circadian clock is it's a timer, and it's a timer that's based on a transcriptional mechanism. The other thing I want to make really clear now, and I will say it throughout the talk probably, is that this timing mechanism is found in all the cells in your body. This is not just a neuroscience project. This is a general physiology project. You know, whether you're a liver or a kidney or a bone or a lung or a skeletal muscle, every single cell in your body has a clock. So this is the cartoon in the bottom is a simplistic drawing of what this transcriptional timekeeping mechanism looks like. So I'm going to start down here in the bottom left with the two proteins. One is called BMAL1. The other one is called clock. These are transcription factors that will heterodimerize, so they'll bind together. And then their target sequences on genes are EBOX elements. And then they will serve to transcriptionally regulate expression of genes in the period family, so per 1, 2, and 3, and the cryptochrome family, the CRYs 1 and 2. And so these messages are obviously moved into the cytoplasm. They're translated into their protein products. Out in the cytoplasm, they can be post-translationally modified by phosphorylation and probably some other ways. These will translocate back into the nucleus, where these proteins will serve to inhibit BMAL clock function. And the dynamics of this process take approximately 24 hours. So this is the timer. And we talk about BMAL clock being part of the positive limb, sort of transcriptionally activating period and cryptochrome, while the PERS and the CRYs comprise what we call the negative limb or the inhibitory limb, as these will come back and inhibit BMAL clock function. Now, if you change the stability of some of these proteins, let's say the PER protein or the CRY protein, you can also change the timing that is used. So there is an example where there's a human mutation in the period 2 gene, and that period 2 protein is less stable. And what that leads to is a shorter period length. So those people have a intrinsic period length of about 22 hours. So the 24-hour period length of this timer is really a combined product of the rates of transcription, the rates of translation, and the stability of the protein products that are involved. All right, so now what I want to transition into is talk about what does the clock in muscle look like. And actually, muscle's a fantastic tissue for circadian rhythms, in part because we can get, obviously there's a lot of muscle. We can get muscle from different rodent models, whether they're rats or mice. But in some cases, and in some places in the world, one can do serial biopsies in a human because we can get access to vastus lateralis. And in fact, circadian collections have been performed in both rodent and in human studies, which has been really helpful for us to try to understand similarities and differences between mice and humans. All right, so what are the criteria for the collections? That's what this sort of cartoon slide is about. So again, we want to do our tissue collection under constant conditions. So generally speaking, for the mouse work, we do things under dark conditions with maybe a little help from red light because the mice do not note the red light. And then we set up the collections for over 24 hours where we collect tissues every 4 hours. All right. And Brianna can share some of her experiences doing that when she was doing her PhD in my lab. And then once we have all these tissues, then what I'm going to talk most about right now is we will isolate RNA from these tissues. And then we originally we used gene arrays, but now we do RNA sequencing. We have particular statistics that we have to run to ask whether things are expressed in a circadian pattern or not. And then that becomes what we call our circadian clock output data or clock data. So here's an example of what gene expression looks like in mouse muscle. Over in this case, these were a series of experiments that were done over 48 hours. So this group really did heroic efforts. So they collected tissues every 2 hours for 48 hours. And so let's just focus on the graph up at the top. This is for BMEL1 messenger RNA. This is starting in the middle of the dark phase. Then, and this is all done under dark conditions. These light dark are just so you have a perspective of where these genes are up or down. So these are collected under constant conditions. And so the expression of BMEL message is highest at the end of the dark phase. It goes down rapidly during the rest phase, goes back up during the dark phase, and then down again. And one thing I'd like to point out is the difference in expression between the lowest point down here and the highest point at the end of the dark phase is on the order of fourfold, not 40%, not, I mean, fourfold. This is a very large difference in message levels. And so depending, even if you don't study circadian rhythms, if you happen to be collecting tissues here versus here, sometimes you can find very different levels of expression just because of time of day effects. So it's something to keep in mind. Just to illustrate the negative limb, here's PER2 messenger RNA, and hopefully you can see it's antiphasic to BMEL. So it is now lowest at the end of the dark phase and highest at the end of the light phase. And so these are two examples of genes that exhibit a circadian pattern of expression. All right. So what we get, one of the questions I have gotten for over 10 years now is that, mice are nocturnal, and in general, humans are not, although maybe some of you are, but in general, most of us aren't. Most of us sleep during the dark phase and we're active during the rest phase. So the question is, how does the clock in muscle in a mouse look compared to the clock in the muscle of humans? All right. And so what I've got in the middle of this figure is some data we put together, my lab and combined with a group from the Netherlands, put together in this review, Ticking for Metabolic Health. We took data from the human muscle circadian data set, which was published in 2018, where they did, they had a cohort of about 10 humans, and they collected muscle biopsies from them every 4 hours over a 24-hour period. And then the data set from muscle was taken from this paper. We did some Z-scoring to normalize so we could sort of line them up. And then the only thing we did that was different is we just are presenting this as active phase versus rest phase for the human, and active phase versus rest phase for the mouse. And hopefully, without getting into too much detail, what you can see is the pattern of BMEL and period 2 are virtually the same in the mouse and the human skeletal muscle, as long as we look at the data in terms of active and rest cycle. Obviously, if you look only at light and dark, you get a different picture. But the way we think about it now is that the clock and muscle actually between mouse and human is actually quite similar, as long as we keep our reference point being the active phase and the rest phase. All right, so I've talked to you about the timer, but this is a physiology society, and I'm a physiologist. And so sort of like, and as when I first started in this field, I had a number of people think that I was crazy because, well, for various reasons. I'll leave it at that. But the key point with this that I want to share with this slide is the clock is more than a timekeeper. And that's why this is important for physiology and for physiologists. So what I've got on this slide is sort of an expanded clock function. So up under the sort of in the lighter images, It's the blue and green box are clock and BMAL. So this is what I've shown you before. They will regulate period and cryptochrome. These, get translated into proteins where they translocate back, and then they inhibit BMAL clock function. So that's the timing mechanism. That's the same. That's in every cell. Here's the important part. It's what we call clock output. It turns out that the BMAL clock transcription factors are actually quite active outside of their timing mechanism. And they will bind to thousands of genes across the chromatin. And through this action, they will link timing, clock timing, to daily physiology in a cell. And so I just want to say, even before we get into some things, is that there are a number of labs now that have published across lots of different cell types that there are time of day differences in cell pathways. Metabolism is probably the most well studied, but we can see that. So let me take a step back. So what I want you to walk away with in this slide is that there's some physiologies within your cell of interest, within your tissue of interest, that are actually oscillating over time of day. So I'll talk later about some of Brianna Harfman's work, in which she showed her data implicate substrate metabolism as something that the muscle clock is regulating over time of day. You can go in whatever your cell of interest, and you will find that there are some cell functions that are very much under clock control. So if we go back and say, okay, well, what does the circadian output look like in skeletal muscle? So this was work, again, it's been about 7 years or so that we published this. So what I'm going to talk about here is that the daily program, or that clock output in skeletal muscle, includes greater than 1,200 genes. Now, so we collected these tissues, we did this expression. So this is a heat map over on the left side. The yellow highlights the time at which these genes are peaked. So what we've done is we've clustered those genes that peak together at this time, peak here, peak here, peak here, and so forth, okay? And as you can see, the biggest population of genes that are peaking is here in the middle of the active phase in muscle. And then I've just, and the table that's just to the right are the genes involved, are the genes that are in this heat map are involved in the following functional categories. So transcriptional regulation, there's a lot of transcription factors in this, MyoD1 being one of those, and that's something that my lab has a lot of interest in. Another one that's been important in the muscle and the muscle atrophy field is the E3 ligase atrogen. So this one is also regulated in a circadian manner. And then for people that really think about muscle mechanics, stiffness, there's a gene that is downstream of the clock called RBM20, and this is a gene that targets titin for splicings. And then you can find a variety of other nutrient and exercise signaling pathways, like the AMP kinase pathway, amino acid transport, lipid metabolism, and one of my favorites, the Z-disc, has a lot of circadian regulation, and that has implications for sarcomere structure. All right, so that hopefully gave you a brief or a background in kind of what the clock is and what are some of the clues that we have in terms of what it's doing in muscle. So then the next section of this talk is going to be about, does it really matter? And again, this sort of, it goes back to when I first started in the circadian field. I had some of my colleagues talk to me and say, look, this is sure, these genes go up and down, but really it doesn't matter. It's not going to affect the physiology, or it's not going to affect the exercise. And so, one theme of the research in my lab then had to focus on actually asking this question. And so we had to ask, if we disrupt the clock only in muscle, does it actually affect the physiology? And so, can we get some data that would suggest that this really is important? And so to do this, we had to make a mouse. And so the way we did this is, so we wanted to stop the clock or knock out BMAL, one of the core. So the gene we're going to target is BMAL, which is this one down in the bottom left that I have circled in red. And we're going to do it genetically. So we're going to take a mouse that has the gene B-male floxed. And then this was a mouse we actually made at the University of Kentucky when I was there, which is the human skeletal actin promoter, which drives a tamoxifen sensitive Cre recombinase. So by getting these crossed mice, what this allowed us to do is wait until the animal was an adult, meaning 12 to 14 weeks of age. We would then treat it for 5 days with tamoxifen, which would induce loss of the B-male locus. And thereby we could do this in a mouse. We're not going to affect the central clock. We're not going to affect the clock in other tissues. We're only going to affect the clock in skeletal muscle. And within skeletal muscle, we're only going to affect it in the fibers, not any of the other cell types like satellite cells or something like that. Then there's a whole bunch of, PCR gels and some Western blots just to say we did it. And it was very specific to skeletal muscle and all skeletal muscle. And we did not see any recombination in heart or any other type of tissue. So to start with, just a few slides ago, I had showed you the heat map for control mice, and that's this one on the left side. So these are mice that are the same age. And then the cohort of mice that lost B-male only in skeletal muscle, we did a time course collection, and the data from this gene expression study is organized here. So what we did for this is we took those genes that are circadian in the normal mouse and then just asked what's the pattern of their expression in this tamoxifen or the B-male knockout mouse. And hopefully what should be obvious to you is that we lose circadian patterns. Now, it's not simple. It's not like everything turns off or everything turns on. It's just really noisy and the pattern is non-specific. And I also want to point out, we did monitor their feeding and activity patterns, and there's no change. So this significant change in transcription or transcript levels in the muscle is really due to the loss of B-male and not due to some behavioral change. All right, so I've always been sort of interested in muscle structure function. So really one of the first things we asked is, how does this affect muscle function? And so we did this in 2 ways. So in this top figure here, mice are really handy. When you hold them by their tail, they will grab onto wire mesh, and you can stick that on a force transducer and measure their grip strength. All right. And so that's what this data is here. We looked at grip strength in the wild type animal and then the age matched muscle specific B-male knockout. And you have almost a 20% decline in grip strength. And so that was promising and that was exciting because again, this was a muscle specific knockout, but grip strength is a neuromuscular measure. So we needed to do the sort of what I would call gold standard for muscle force. And that's the ex vivo muscle mechanics test. So in this case, what you're seeing in this blown up image is a mouse muscle in a bath, and it's tied at both ends. One end is a force transducer, and the other end is just fixed. And we're going to go in and with electrodes, and we're going to stimulate the muscle at different frequencies, and then measure the amount of force that gets generated. And the data from this assay is down here in the bottom in what we call a force. So force on the y-axis, frequency on the x-axis. As you would expect, at very low to low frequencies, this is below the threshold. So we're seeing very little force on this muscle. But as you increase the frequency, you increase force generation. Up to a point you get into a tetanic, well, these are isometric. So, but you get a maximum force generation. And so here's what we get in the wild type animals. And what we see in the B-male knockout mice is we have about a 15 to 20% reduction in maximum isometric tetanic force. And hopefully, if you've noticed, we actually normalize our force to muscle size. So this is not a muscle atrophy problem. This is actually an intrinsic capacity for that muscle to generate force. And because we do electrical stimulation, this is independent of innervation. So if there is something that had gone on with the neuromuscular junction, the motor neuron, this is independent of that. So this is how I think of it when I talk about muscles being weaker. We have its maximum isometric force is down. This is taken into account the size, and it's done in a way that is independent of innervation. And so now we get to go into some of Brianna's work, although I did the faux coloring on some of these bars. I apologize, Brianna. But one of the things that Brianna was really quite interested in was the metabolic effect on what that happened when we stopped BMAL. And so what she found in her dissertation work was that stopping the muscle clock leads to altered glucose homeostasis in the whole animal. So what she did is she took the cohort of mice, she fasted them, and then did blood glucose measures. And actually, if you just look at fasted glucose, it's really not, obviously there's no change. So that seems like, okay, fine. But she was smart enough that she actually took that blood and did a measurement of insulin. And this was what was really striking. It's like, wow, there's something's going on here, because now we've got, on the order of up to 4-fold difference in insulin levels, yet their fasting glucose levels were the same. So clearly in the muscle-specific BMAL knockout, there is more need for insulin to help get glucose into the skeletal muscle. So then she followed this up with performing a glucose tolerance test in these mice. So again, the blacks will be the wild type animals, and the reds will be the BMAL knockout, muscle-specific BMAL knockout animals. So in this case, you take a mouse, you just do an IP injection of glucose, and then do collections of blood glucose at different times after the glucose ingestion, and measure how high it gets and how quickly it takes to disappear. And hopefully what you'll see is in the wild type, you get that quick response of glucose going up, but then it comes down quite quickly. In the muscle-specific BMAL knockout, the excursion of glucose after the glucose treatment is much higher, and then it takes longer for it to come down. And this is summarized down here in the area under the curve. So we have a significant increase in the area under the curve after a single glucose tolerance test, all right? And one piece of data that is always, and I don't know if Brianna remembers this, but she also did an insulin tolerance test. And the data are not here, but I keep thinking about it because it's something we probably should go back and revisit. But when she did the insulin tolerance test, the muscle-specific BMAL mice didn't handle it very well. And so therefore, we had to end the test and obviously not publish the data. But there is something going on between muscle, liver, and the other organs, because the response to insulin was really quite dramatic. All right, back to the story about glucose uptake. So then the next thing she did is what we did, speaking of Michigan, is we contacted a colleague, friend, Greg Carty up at the University of Michigan, because his lab has always had significant expertise in measuring rates of glucose uptake. And so Brianna learned this assay and performed the ex vivo glucose uptake assays. So what this involved is taking the muscle tissue out of the animal, incubating it with a radio-labeled glucose, and then she did it either in the absence or presence of insulin, which is this graph on the left side of your screen. And we got very expected responses. Obviously, muscle tissue is very insulin sensitive, so you see a 2-fold increase in glucose uptake in the wild type muscle. But in the absence of BMAL, she saw no change. So there is some basal glucose uptake. So the muscles do take up some glucose, but there was absolutely no increase in response to insulin. So then she said, okay, well, you can also affect glucose uptake through contractions, and you can mimic this by using ACAR, sort of an AMP kinase trigger. And so again, with the wild type muscles, we got expected results. So we see an increase in glucose uptake in response to ACAR treatment. But like we saw with insulin, there is absolutely no change in glucose uptake in response to ACAR. So this was really quite striking, is that because, the loss of BMAL has led to these muscles not being able to respond to either an insulin cue or a contraction AMP kinase ACAR cue. So that would suggest that the ability of the muscle to access glucose for contractions likely is significantly impaired. So, one question was, well, why is glucose uptake down? Well, the story's kind of not well. We'll start here. The main glucose transporter in muscle cells is GLUT4. And so what Brianna did is she probed us both at the message level, so using qPCR, and found that GLUT4 expression in the muscle without BMAL is about 50% reduced. And then running some Western blots, hopefully what you can see is the decrease, the GLUT4 protein levels are down significantly at this point. And so something on the order of about 75%. So this is very consistent. So there is some GLUT4, but the ability to change in response to insulin or in response to ACAR is going to be significantly diminished in these BMAL knockout mice. And then this is work that was done most recently. It's not been published yet, is we've done some more molecular studies. In this case, this is chromatin immunoprecipitation. And we've identified that the GLUT4 gene is actually a target for both BMAL and clock binding. So we see GLUT4 as a major target of the circadian clock. And so to summarize this part of the talk, loss of circadian clock function leads to intrinsic muscle weakness, and that loss of circadian clock impacts glucose uptake. So when you lose it, they're downregulated. And we would suggest that the muscle is forced to rely more on fat with an increase in amino acid use to support TCA cycle. So in the last few minutes here, what I'd like to do is to bring this back into some physiology and talk about how the clocks might be playing a role in the exercise response and potentially have a role in endurance performance. And so, let me see, where are we? Okay. So where we started this project actually was a fairly simple question, asking, does the muscle clock respond to an acute bout of scheduled running? So there had been some ideas that exercise or contractions or running might be playing a role in setting clock time in muscle. So there were some of the people in my lab decided, well, let's just start and ask if this is even possible. So the question asked is, does running at different times of day alter the phase or change the amplitude? In this case, we're going to look at that core clock or that timer in these series of experiments. And to do this set of experiments requires some unique resources. One of them is a mouse. In this mouse, the work that describes this mouse was published in 2004. But what Sally did is she took the enzyme, the coding region for the enzyme for luciferase, so the one that will take substrate and generate light, and she knocked it right into the period 2 locus of the mouse. So now in these mice, when they express period 2, they also express luciferase. And so this is really clever. So because we can take the tissues out of these mice, we can stick them in a dark box with cameras that will collect light. And we can ask, so when we follow light emission, we are actually following PER2 protein expression. So this gives us a really nice way, a bioluminescence assay, to actually ask questions about clock function. And I've put on the right, this is just an example. This is a single fiber that is from these PER2 luciferase mice. And within a fiber is obviously multinucleated. So what you can see is there's light coming from the different nuclei across the length of the fiber. Now, so what the design is going to look like is we're going to run the mice at specific times of day, and their running is going to be on a treadmill, and it's going to be set. Then we'll remove muscle tissues, and we're going to record light emission as a measure of clock function. And so again, here's the outline of the design. And here's some examples of what these bioluminescent tracing. So this is some data from the lung. You can see over 1, 2, 3, 4, 5 days, the rhythmicity of PUR2 luciferase. And then here's an example from the central clock or the SCN over those 5 days. We're going to be looking to see whether that peak is going to change and where it shows up. So we're going to have a 30-minute treadmill run. We're going to do it at 5 different times. And then what happens to the clock? All right. Oops. Sensitive buttons. All right. So this is work that was done by, shared by 2 postdocs, Denise and Chris Wolf. I think, some of the traces are up at the top and it's very colorful. So the mouse that ran in the middle of the light phase or the middle of the rest phase, we saw a shift to being earlier, or what we call a phase advance. When we ran the mouse at the end of the rest phase or the end of the light phase, we saw what we call a phase delay or a shift behind. And then when we ran the mice in the middle of the active phase, we saw no change. And so I'm just going to point out the changes in the peak phase down here in B. So this is, again, work that was published in 2020. So this is a feature of the clock. So what's happening here is we're applying the exact same running. All right. So it's the same bout, the same speed, the same grade, the same duration, but we're doing it at 3 different times of day. And what our clock data is saying is that the clock will respond differently depending on what time of day you run. So if we ask the mouse to run in the middle of its rest phase, it advances the clock forward by over an hour. If we ask the mouse to run at the end of the rest phase, what's happened is we've caused the clock to delay by an hour. And these are very, very characteristic features of what we call a phase response curve, so that the clock will respond differently to a signal, in this case exercise, depending on where in the cycle the clock is. So that was really exciting because that said, well, time of exercise can provide some information and cues to the muscle clock that's going to be quite different, because the SCN clock doesn't respond to these exercise responses. So then we started asking, well, what about training? Can this have an effect on an endurance performance? And so to start with, always important to note the people. So Stuart Hesketh was a postdoc in the lab. This was a pandemic project for Stuart. And so this was something he was able to do. He did, I give him tons of credit. He spent a lot of time alone with the mice in the animal facility training them. But what he had read and what other labs had reported is that if you take untrained mice and you run them either in the early part of the active phase here, so early dark phase, they have a different endurance capacity if they're tested again here later in the dark phase. And so the first thing he did is he just said, okay, does this happen in his hands and does it happen in Florida? And the answer is absolutely yes. So this is it's really quite striking. So the way we're reporting the data for this set of slides is treadmill work done. All right. So what is work done? It's treadmill speed times the grade of the treadmill times the duration or the time on the treadmill. And then also included is the body weight of the mouse. All right. These animals are exactly the same. So the ones that did the afternoon training were the same ones that did the morning training. We obviously split out the training time, so there was no overlap. But, hopefully you can see that the mice, when they run early in the active phase, have about a 60% reduction, or they are about 60% of the endurance that we get in the afternoon runners. And this is incredibly repeatable. I was actually pretty surprised. So then what Stuart said is, okay, if I train the mice early or train late in the active phase, do the mice adapt the same? So the training protocol, these are going to be female mice. They're trained 5 days a week, and they're going to train for 60 minutes. And what is really important is they are going to train at 70% of their time of day max. So that means the mice that are running in the morning are at the same relative workload, but actually a lower absolute workload. All right. So what happens? So this is just some data from, so that again, this includes the pilot data that's here. This is before they started training. So we still see, Stuart just didn't believe it. So we still see that difference. And then after 3 weeks of training, what you can see is each group is improving. So there is, as you would expect, there's an improvement in endurance performance, but the difference between the late runners and the early runners looks virtually the same. But something happens. So somewhere between 3 and 6 weeks, there's a change in the rate of adaptation. So by 6 weeks, there's actually no statistical difference in the endurance capacity between the morning versus the afternoon runners. And, you know, on one hand, it was like, okay, good, everybody adapted. But then the more you thought about it, it's like, there's more to this. And so what does this mean? So both time of training groups adapt. This is good and this is expected. However, the early runners increase their endurance capacity more, about 1.8-fold from where they started to where they finished at 6 weeks, compared to the late runners. And this is statistically different. So what we can conclude with this is that those mice that were training in the early active phase actually had a greater performance adaptation. And when we stopped at 6 weeks, so, you know, what would happen if we extend the study? We don't know that. And obviously, yes, this is in mice, but we're really pretty excited about that, about this finding. So how or why? What's different in the early versus the late runners? And if you're hoping that I'm going to be able to tell you exactly what it is, I'll just give you an early warning. We still are not sure. So so we did look at some of the simple things. So what we saw is that changes. So is there a body weight change, or maybe there's body composition changes? And what we found is there's a trend for a decline in body fat for those. So the black bars are all the control animals, the blue are the early morning runners, and the gray are the late afternoon runners. And so the body composition is not different. What about some index of metabolism? So we did blood glucose response post-exercise test, and also blood lactate response post-exercise test. And hopefully what you can see is blood glucose goes up after an exercise test. It doesn't change with training, and there's no difference between the groups. And the exact same is true for the lactate. So clearly we were exercising them intense enough, but there really was no sign that there were any metabolic markers that were different. You know, glycogen storage, obviously glycogen is critical for this. As you can see, so the C is the liver glycogen and D is muscle glycogen. We see a training effect on glycogen storage, but we do not see a morning versus afternoon exercise effect. So none of this actually can explain the performance differences. So we went and we asked about whether the changes in the muscle clocks or any of the tissue clocks were affected. So we went back and we took our per-2 loop mice. We trained them for 6 weeks. And then what's really important is we waited 3 days after the last bout of collections, because we did not want to look at an acute response. We wanted to see whether the steady state function of the clock was changing. And what we found is that the muscle clocks shifted significantly in phase, and the direction of the shift depended on the time of training. So just like with our acute exercise. And I do want to point out, there is no change in the phase of the central clock in the SEN. So what we saw with the early runners is we saw a really significant, I mean, we're talking 3 to 4 hour shift in the muscle clock to an earlier time. The late runners, we saw a shift in the other direction. So the runners are in red, the controls are in black. And so what this is saying to us is that the muscle clocks are adapting to the time of training. And we view this as that the clocks actually, so the exercise is a cue. And so the clocks in the muscle are shifting so that they are better aligned with the exercise. So that, and we propose that this is actually a contributing factor to the time of day exercise performance. Do we see shifts in other tissues, we do see some, but not as dramatic. So I've just shown some both lung and white adipose tissue clock shift. The early morning runners, those lung and adipose do not shift. Late runners, we do see a delay in those. So we also think that there probably are some systemic effects in terms of exercise performance as well. So the take-home points. Muscle clocks are sensitive to time of training. Remember, clocks change, so that whenever you change the clock, the timing of the clock output and the timing of the physiology and the timing of the metabolism will change. We propose that training-induced shifts in muscle clocks are required for enhanced early morning endurance adaptations. The central clock does not change, but lung and white fat clocks do. This suggests but they only do it with the late training group. This suggests maybe exercise has an indirect effect on the clocks in the lung and the white fat. And lastly, our work highlights the potential to use scheduled exercise as a therapeutic strategy to enhance clock function and muscle health. We would also say that, this could be important for sports performance as well. But I think basically athletes already do this. So they actually generally train at times they're going to be competing. Last but not least is obviously the acknowledgement of all the people. It's been a fun time. We miss Brianna, but there are a lot of fun people around. So thank you very much.
I missed the lab as well, but I'm sure you always have a fun lab because you foster that kind of environment. So thank you very much for that talk. I want to open it up for questions now, and we were going to let people know that if they want to raise their hand, I can call on people, or Dr. Esser can call on people, or if you want to put things in the chat, I can read them out as well. And we have had one question in the chat for a few minutes, so I'm going to go ahead and ask that one first, if that's all right.
Mm-hmm.
So Kevin wanted to ask, so fascinating earlier slide, someone with a circadian sleep disorder could potentially be at higher risk of diabetes. So is that true?
Yeah, so actually that's a great point. I didn't, so there actually are a number of studies now that have taken, the one I think of sort of at the top of my head was probably published around 2006. So a group in Harvard took, young, healthy people, and then, well, actually it was males, and then put them on a 28-hour day. So they had them come into their clinic and had them live in a 28-hour day, which meant they were 14 hours light, 14 hours dark. And after a week of that environment, they exhibited hyperglycemia and their mean arterial blood pressure went up. So that was really the first data in humans that would suggest that indicated that disruption of the circadian system leads to sort of a pre-diabetic phenotype. But there have been studies since then. So putting people on shift work, but getting back to the question, which was about disrupted sleep, absolutely. So these all fit under the category of anytime you disrupt sleep, whether it's just sort of intrinsic properties of the person or whether it's shift work or whatever, then there are absolutely circadian disturbances, and almost always these come with kind of a pre-diabetic phenotype. And where we come in, and I have some colleagues in the Netherlands that are doing this, is that we would suggest that muscle is a big part of that. It may not be the only part, but the disruption of the muscle clock has just profound effects on its ability to use glucose. And so anytime you affect muscle and glucose, you have a systemic glucose handling problem. And so we do, yes. So the bottom line is, yes, I think there's an association there, and it's going to be really important to understand better.
Thank you. I think Naveen has a question next.
Yeah, Dr. Esser, thank you very much. I'm Naveen Sharma from Central Michigan University. I think in one of those 1 degree of separation things, I spent my postdoc in a few years with Greg Cartier at Michigan, and I left in 2013. So I definitely know that glucose uptake assay very well. So I was going to ask regarding those muscle-specific BAM1 knockouts. It was interesting, right? When ACAR was unable to produce any, further or help really rescue those animals. But did you end up look at, and you noticed that GLUT4 was down, but was there any other detriment or I guess impact that both contraction or insulin signaling pathway. So were AMPK levels down as well too? Because I think you mentioned that some of those genes from those knockouts, AMPK related genes were down, right? So I would assume that.
Yeah. So in part, I want to say, hey, Brianna, talk about your dissertation. No, but I, so what can I tell you? We haven't really looked at the acute response, whether it's sort of AKT, AMP kinase, sort of signaling pathways. What I can tell you is that it's and I started to, but then I realized I was going down a bad hole. Was that it's more than GLUT4. So one of the things that Brianna found in her study were a lot of the enzymes in glycolysis and key ones, hexokinase, phosphofructokinase. The expression of those genes were down. The expression of those proteins were down. The enzyme activity of those proteins were down. And so that one, so what I think, so I'll defer a little bit to Brianna, but what my understanding was when we finished up her work is that clearly there's a GLUT4 effect, and whether the lack of response to insulin or ACAR was just there wasn't more GLUT4 to actually translocate to the membrane, whether that was due to signaling pathways not working, or, the whole system was kind of stuffed up. So we did some targeted metabolomics with a group at Michigan, and actually in those muscles, glycogen levels are higher. But what we see is that pathway looks like flux through glycolysis is way, way down. And so there's also kind of what I would imagine kind of a concentration-based backlog in terms of getting stuff in. Mm-hmm. And to be honest with you, after she left, we really haven't pushed this too much. So we don't know much more about that. The questions you're asking are really important and things we've thought about. It's just haven't gotten there.
No problem. Good reason to talk to Brianna on the side too. That's good.
Yeah. We'll jump in and just say, I agree with your interpretation that it did seem like the flux was really down. I think because my focus was more on the insulin, I didn't look as much at the AMPK pathway and looked at where they kind of came together, but we did do some work with TBC1D1 and TBC1D4 and trying at the beginning of translocation. So I'm happy to talk about that with you.
Yeah, that's a good point. I think it was TBC1D4 was way down, if I remember correctly.
Yes.
So yeah.
All right. Thank you very much.
Mm-hmm.
We do have another question in the chat. It's from Alex Montoy. Heart attack risk is highest in the morning in clinical populations. How can we balance this risk with the potential for more effective exercise training from morning exercise that you've shown?
Very good question. So yeah, I mean, when you look at the big population data, the probability of a cardiac event is going to be higher in the morning than it is in the afternoon, for sure. And in fact, you can dig into the data. So I think Michigan still does spring forward kind of things, right? You do that unfortunate change in time. If you actually take that day after spring forward, that's also one of the biggest days in the ED for cardiac events. So this idea that you're messing with this, you're messing with the circadian system. Now, that said, I think so I don't think the risk of the cardiovascular problems outweigh the benefits of the exercise. I mean, this is a great, I've never had this question. So that's a great question. And I think from my understanding of the circadian system and the circadian heart and adrenergic system, it's just if you have, it's going to depend on the age of your person. So the younger, the better, the older, the worse. So I would, if it's an older person, I wouldn't want them to hop out of bed and jump on a treadmill at 80% of max, which I don't think most people would suggest anyways. So for older people, I would say, make sure if they're going to start training in the morning, that they, at least initially until they get trained, that they train, they ease into that training. I think once they've trained for a while, my working assumption is your clocks are, one of the things that exercise is doing is it's synchronizing clocks in the system to that training time. And so that then you can handle that increase in intensity and stuff that would come. But I mean, the probability, so I still think that the benefits of exercise far outweigh the risks in the morning. And if that's the time you have a person that can and will do exercise, by all means, find a way to help them do it safely.
Great, thank you. I'm going to turn it over to Karyn Ball.
Yeah, Karyn, in showing the data on the glycogen levels, there were no difference between the early and the late runners, late trainers on muscle glycogen, but you noted that the liver glycogen was higher in the early trainers. What do you make of that? Does that suggest some sort of sparing mechanism? Are you thinking of doing any more with lipid metabolism assessment?
Yeah, no, it's a great question. Because I think, there's this intersection between sort of feeding behaviors, morning exercise, afternoon, sort of this combination. So I think liver glycogen's important. I think it's critically important for an endurance event. And so one of the things we are about to start, so we now have some cages, which Brianna would have loved when she was here, that actually we can control and monitor feeding. So we will know exactly when they eat, we will know exactly how much they eat. And so we will have all that data then to bring into this. So from a speculation perspective, I think the liver clock is changing as well. And I think the liver clock plays a role in, so what we know with, which makes sense, during nighttime, liver glycogen goes down as the liver is working to maintain blood glucose during that long, assuming you're not eating in the middle of your sleep, during that long fasting period while you're sleeping. And then glycogen gets stored throughout the day. I think the clock plays a role in that in the liver. And I think I'm going to guess that there's some interaction between what's going on in the muscle, what's going on in the liver, what's going on in the adipose tissue. And yes, I think that it would be really interesting to also couple this with lipid availability, because again, sort of Brianna's ugly twin, Brian, had done some work that would suggest that oxidative metabolism in muscle is also modulated by the clock. And so one would expect that would be, and so then availability and the ability to use lipids is likely going to be time dependent as well. So you raised some really good questions. If you guys have some students that might be interested in pursuing those kinds of things, have them feel free to think about applying to University of Florida's graduate program.
Thanks, Karyn.
I do see more hands. So I'm going to get to you. I'm going to jump really quick because you mentioned the metabolic, the oxidative changes in the metabolic flexibility. When you were showing the differences in endurance between the morning runners and the afternoon runners, I felt that was really interesting. Because if I remember correctly, the fat metabolism enzymes were higher in the early morning, which I would think would be when you'd have more endurance. So I'd be kind of curious to look at which enzymes are higher and how that's kind of correlating.
Yeah, so what you're remembering are transcript levels. So you might want to put a delay on those because
Gotcha.
yeah, absolutely. But yeah, the concept that we had with Brian's work was that the clock was basically turning on genes important for fat metabolism at that transition of before they transitioned into the active phase. But in terms of tracking protein amounts and that, I think that the devil's in the details there, but you're absolutely right in your memory.
I'm going to turn it over to Jenny Branish now. She's here now.
Thanks, Karyn. So just because you mentioned daylight savings time, as a parent whose child never seems to want to adjust to such time changes, now I'm really curious what your sort of professional opinion is on all this talk of us
It's really
eliminating daylight savings time.
So we shouldn't, there's no reason to change time at all anymore. And I mean, and there's health reasons not to. And then we should stick on standard, not daylight savings. So there's actually quite a bit, and this is something the Society for Rhythms, Biological, SRBR, Rhythm, Society for Research in Biological Rhythms, has been very active in this space in terms of lobbying the legislature, because there's been all this push, let's keep light later. Well, I mean, that's summertime talk. Just wait till, so we could go on, no time change and go on standard, be daylight savings time only. And it's going to be dark, and your kids going to school are going to hardly see any daylight, you know? Anyway, but there's quite a bit of research on this. So if either you as an educator or a scientist or a parent are interested in information, their website has available information on this, and Brianna can point you to the website if you want.
Yes, I can. Charles.
Karyn, I think I'm probably going to pull together a lot of different comments and questions into one. Well, I'll summarize it as I'm interested. I think I'm interested in what's going to happen with insulin receptors, because you
That
talked about
caught
RBM.
me off guard. You caught me off guard with that.
So you talked about RBM, and I know you've got the paper. I think it's Melanie left this paper in the email, Global Knockout, that shows sarcomeric protein changes. So RBM is downstream of insulin receptor also.
Oh, interesting.
RBM20 is.
Okay.
And it's probably also signaling through some estrogen receptors. So both insulin and estrogen might come into play, but you haven't talked about the sex differences. And I'm sure I can probe Brianna's mind later about insulin receptor, but what's your take currently on the sex differences, especially in the muscles?
Yeah, so I mean, so first off, what I can tell you is, so it's interesting. I didn't know RBM20 was downstream of the insulin receptor. That said, we do have, so we've got Lance Riley's manuscript that I was talking about that's still, it's in revision. We're about to get those comments back. But so we've identified that RBM20 is actually a direct target of the clock. So it may be still targeted by insulin receptor, but the clock is part of that regulation anyways. Sex differences, we know very, very, very little, like zero. But so part of the problem has been the cost. So if you're going to do a circadian analysis, where you're collecting tissues over 48 hours, and then you're going to do RNA sequencing and stuff like that, it gets costs go way up very fast. That said, I'm working with Brian Delisle. Some of you know Brian down at University of Kentucky. And so we got some funding to do male and female mouse hearts. And we'll probably plug some muscles in there as well, because I think there are going to be differences. I think there'll be more overlap between males and females than, let's say skeletal muscle from a male, skeletal muscle from a female. There'll be some overlaps there. Whereas if you take within a male and compare a skeletal muscle to a heart, there's next to no overlap. It's just the clock genes are the only things. So I think there's going to be more overlap between the sexes than between the tissues. But there's a lot to learn in that space. And so we're really excited to have the opportunity to actually generate that data. And it will be, as soon as we have it, it will be made freely available on our website. So because I think, obviously, as we start looking at sex as a biological variable, this is going to be important.
Thanks, Karyn. Good to see you again.
It's good to see you too.
Do we have time for one more question, Steve?
Yeah, hi Karyn, this is Steve Elmer from Michigan Tech. A wonderful presentation, and thank you for your willingness to record it. I know there's several grad students that will really enjoy watching this afterwards. My question is related to the muscle weakness data that you showed. And if I follow right, you reported about a 20% reduction, specifically in isometric force. And when I think of human movement, isometric force is really important. Most movements we do throughout the day are dynamic in nature, and so we also need to consider not only the force generating capacity, but also the shortening velocity. And so I'm just wondering, has your lab or other colleagues out there looked at this this effect with the muscle clock on dynamic muscle function? And if not, would you expect that there might even be a greater reduction in the dynamic muscle function compared to the isometric?
Yeah, no, I mean, it's something I think about a lot, because I refer to isometric because it's just a way to standardize. Once you start getting into dynamic measures, then things can get murky really fast, at least in my perspective. So I do, we don't have any data. And when I, go to and that's when actually I had over the pandemic, one of the graduate students did a review on this time of day strength difference. And there's a lot of work out there in that space but we ended up having to focus just on isometric because the dynamic stuff, people do things differently. So we couldn't, it was just, we were unable to compare across. But nobody's really, as far as I can tell, nobody's really addressed this from a very focused perspective. I would imagine, I mean, you know, do we see any sort of myosin heavy chain effects? Not really. So I don't see a major shift in, you know, maximum velocity of shortening, sort of. But could there be effects? But there are other parts of the contractile elements. So there's a light chain kinase that's affected. So part of that is we don't know exactly what that light chain kinase is doing, but assuming that it's targeting a light chain, could it affect potentiation and some other aspects of the twitch? Yeah, it could. And some of these, so again, we look at things at the message level, but some of these have proteins that are actually oscillating over time of day. So we've actually looked at some of the proteins, and so there is room for this to be a real thing. We just haven't done that yet.
Thank you.
Mm-hmm.
Well, we have more questions, but unfortunately we are running out of time and we need to transition over to our next event. I will probably be reaching out though, because I would love to talk about the insulin experiments that you mentioned. I do remember what happened to those mice. And ask you a couple of these other questions later on.
Yeah, and if anybody, so how about if I put, I'm going to put my email address in the chat.
That would be wonderful. And I would
So if anybody wants to ask any questions or contact me, just don't hesitate. You might need to say, okay, I was, you know, just put it in some kind of frame of reference like the Michigan Physiological Society. But I'd be happy to talk to anybody afterwards.
Wonderful. And thank you. I want to thank you on behalf of the MPS. I would also like to say this is a fantastic talk, although I am very biased. And I also want to take a moment to just recognize you because not many people probably know this, but we do provide our keynote speaker with an honorarium, and Dr. Esser actually generously donated her honorarium back to MPS to support student training. So that was very.
That was happy. This is a small thing, and just I am excited about the next generation of scientists and what you guys. So those of you in the faculty positions, thank you for all the things you do, and those of you that are trainees, you know, be curious and follow your interests. So congratulations to you all.
We really appreciate you joining us today, Karyn.
Mm-hmm.